DNA or deoxyribonucleic acid is the blueprint of life. It contains the genetic instructions that determine an organism's traits and characteristics. But how does DNA replicate and transfer its information to create proteins? This is where the central dogma comes into play. The central dogma describes the flow of genetic information within a biological system. It states that DNA is transcribed into RNA, which is then translated into proteins. Let's take a closer look at the steps involved in DNA replication.
The first step is to unwind the DNA double helix using enzymes called helicases. Once the strands are separated, primers attach to the template strand, which will serve as a starting point for DNA polymerase to begin replicating the DNA. Each nucleotide consists of a sugar, a phosphate group, and a nitrogenous base. The nitrogenous bases on the template strand serve as a guide for complementary base pairing, adenine pairs with thymine, and guanine pairs with cytosine.
As DNA polymerase moves along the template strand, it adds nucleotides to the growing strand in the 5' to 3' direction. This creates a new DNA strand that is complementary to the template strand. The end result is two identical copies of DNA, each consisting of one old and one new strand. The next step in the central dogma is transcription, where DNA is transcribed into RNA. In this process, RNA polymerase unwinds the DNA double helix and creates a complementary RNA strand using the template strand as a guide. The resulting mRNA strand consists of a sequence of nucleotides that is complementary to the DNA template strand. However, instead of thymine, mRNA contains uracil as a complementary base for adenine.
The final step in central dogma is translation, where mRNA is translated into proteins. This process occurs in ribosomes, where tRNA molecules bring amino acids to the ribosome based on the codons on the mRNA strand. tRNA molecules have an anticodon that is complementary to the codon on the mRNA strand, allowing them to bring the correct amino acid to the ribosome. The amino acids are then linked together to form a polypeptide chain, which folds into a specific shape to create a protein. Based on the nucleotide sequences, the proteins that will produce from mRNA are
And that's the central dogma of molecular biology, from DNA to RNA to proteins. This process is the foundation of life as we know it.